patterns in a developing organism. But we've mostly skipped over the details of the specific molecular mechanisms by which such factors actually influence the behavior of cells. Now, we know that signals must be involved in turning on and off particular genes in cells, genes that ultimately determine how a cell will differentiate, that is, control that cell's developmental fate. What we're going to do in this last lecture on development is to look at pattern formation in an organism in which the genetic mechanisms underlying development are fairly well understood. The organism we'll look at is the fruit fly, Drosophila, an organism we encountered earlier in our course when we talked about genetics. Now, Drosophila has proven to be an especially good model system for studying development precisely because its genetics are so well known. And as you'll recall, the life cycle of Drosophila takes only a couple of weeks. These organisms are cheap, easy to maintain in the laboratory, all advantages that were recognized by Thomas Hunt Morgan when he first established Drosophila as a model for genetics 100 years ago. Now, our goal today is not to focus on Drosophila for its own sake, but instead to use this example as a way to illustrate how gene expression is controlled during development, which will tie together many of the themes we've encountered over the last several lectures. First, we have to talk a little bit about the biology of Drosophila to get us started. You see, although Drosophila is a great model organism in many ways, it does have one little difficulty as a model for development, which is that its very earliest developmental stages differ in some respects from what we've seen in organisms like sea urchins and frogs. Specifically, and I'll describe this in more detail in a moment, the first rounds of mitosis associated with cleavage work in a very different way in Drosophila. So we need to spend just a bit of time looking both at the body plan of Drosophila, in other words, where we need to go with developing a Drosophila, and at this pattern of development. Now, let's start with body plan. Fruit flies, like all insects, are very highly segmented animals. By this I mean that their bodies have an obvious modular construction. If you look closely at an insect, you'll see that it has what look like separate compartments running along the axis from the head to tail. These compartments or segments are easier to see in some insects than in others. They're especially easy to see in the abdomens of adult insects, and they're particularly apparent in the juvenile forms of insects, which we generally call larvae. Now, the larval form of the fruit fly is a little worm-like grub with obvious rings marking the segments along its head-to-tail axis. When the larva fruit fly later develops into an adult, these segments go on to become specialized into different parts of the animal, such as different regions of the head, regions of the middle part of the body called the thorax, or regions of the abdomen. The point here is that segmentation sets up the eventual functional differences that develop along the body axis from head to tail. Now, because insects are obviously segmented, the development of these segments provides a very useful case study in pattern development. And the question we really want to ask is, how does a segmented structure with an obvious head-to-tail axis arise from a uniform, non-segmented beginning, such as a zygote? Now, as I said a moment ago, the very earliest developmental stages of Drosophila differ somewhat from what we've seen so far. Similar to what we've seen, fertilization creates a zygote, and following fertilization, the nucleus of the zygote begins to divide rapidly. There are a number of mitotic events, with the nucleus replicating the DNA and having that DNA separate out into two equal components. But what's different here is that following that replication and mitosis, cytokinesis doesn't occur. Remember, cytokinesis is the last phase of normal mitosis where the two cells eventually pinch apart, creating two separate structures. But in early cleavage in Drosophila, we don't see cytokinesis. So what essentially happens is that you start with what you would picture as sort of an oval-shaped zygote with a single nucleus, and then after about 10 rounds of division or so, we still have just one cell that's got several thousand nuclei in it. Now, what happens next is that these nuclei all migrate to the outer surfaces of this zygote, so that we now have an oval-shaped zygote that has these thousands of nuclei all sort of lining the outer side of it. I mean, on the inside, but sort of leaving the center of it free of nuclei. Then, at about the 13th round of division, all of a sudden cell membranes appear that segregate all of these nuclei off into their own little cells. And what we're left with is essentially an embryo that is in the blastula stage, a hollow ball of cells, in this case all of the cells being formed essentially suddenly with this 13th division, each with its own nucleus, and on the inside is the remainder of material that we would call yolk, which will nourish the developing embryo. Okay, now to anticipate where we're going, the zygote, as you would imagine, already has an asymmetric distribution of cytoplasmic determinants. After the blastula has been formed by membranes forming around all of these nuclei, any asymmetric distribution of these cytoplasmic determinants in the zygote will be reflected in differences among these cells, because cells will have the little piece of cytoplasm that was occurring along the axis wherever that nucleus happened to be. So cells that are appearing at what will become the front end of the embryo will have a little piece of cytoplasm segregated off from that part of the zygote. Cells at the tail end will have a little piece from the tail. So we're preserving the asymmetry that was existing earlier in the zygote, but in a very different way than what we saw previously. Okay, now after we have this blastula, Drosophila, as you can imagine, next undergoes gastrulation, and we don't really need to worry about the details of how gastrulation occurs in Drosophila. But what is important is that as the embryo develops beyond this stage, the body segments I spoke about earlier begin to appear, with obvious differences between the head and the tail ends. At first, these segments all look identical. In other words, you will see the developing embryo just start to develop little rings that are marking where the segments are. But eventually, as I said before, those segments will develop into very different parts of the animal. And in fact, even adjacent segments can be quite different at the end point of development. In Drosophila, for example, one segment near the middle of the animal, say, let's call it the first thoracic segment, will eventually end up developing a pair of legs. But the next segment back, the second thoracic segment, will end up developing both a pair of legs and a pair of wings. So these segments define little compartments which then go on to have different developmental fates. Now, given this pattern of development in Drosophila, as I just described, we're left with two related questions concerning pattern formation. The first question is, how do we get a segmented structure from an unsegmented one? And the second question is, how do segments at the different points along the head-to-tail axis come to develop into different kinds of structures appropriate for their position along that axis? The answer to both of these questions, as you might guess by now, involves the differential expression of genes. There are many genes that control the emergence of the anterior-posterior axis and segmentation in Drosophila embryos. The majority of these genes, and this is the point, are regulatory genes. That is, their gene products are transcription factors which will then turn on and off the expression of other genes. These genes are expressed in a sequential, cascading fashion, essentially building out of the original asymmetry of the zygote, the ultimate asymmetry and structure of the developed adult. And that's the take-home message that we're going to try to get to by the end of this lecture. So what I'd like to do now is to look in a little more detail at part of this story to give you an idea about how gene products control the development of the head-to-tail axis and segmentation. In the 1970s, two developmental geneticists, Christiane Nusslein-Volhart and Eric Wieschaus, set out to identify the genes that are involved in pattern formation in Drosophila. They used a technique called a mutant screen in which they exposed Drosophila to treatments that damage DNA, cause mutations. And then they asked which of these mutations appeared to affect pattern formation in any way. Now, we saw an example of a mutant screen earlier in the course when we talked about the work of Jacob and Monod as they were trying to understand how gene regulation worked in the lac operon system of bacteria. I told you then that mutant screens are extremely laborious because you're essentially looking for a needle in a haystack. You're saying, give me a mutant and I'll see if it affects the particular function I'm interested in studying. But in the case of Drosophila, for example, there are thought to be about 13,000 functional genes in the Drosophila's genome. And Nusslein-Volhard and Wieschaus set out to try to identify every one of the genes of those 13,000 that affected pattern formation in any way. This was actually made especially difficult for them because they were interested in development. And you'd expect that the mutations that arise that are influencing development would very likely be lethal, because if it influences some stage early on in development, it's unlikely the embryo is going to be able to go on to develop into an adult. So to study these kinds of mutations, they actually had to do genetic screens with recessive mutation, or genetic crosses with recessive mutations, and then look at in other words, the adult animals had to have the recessive gene to stay alive. They would cross those, and then they would look at what the different kinds of embryos that eventually died looked like and try to infer how that particular gene might have affected pattern development. A very laborious process and a massive effort. But it turns out, eventually they succeeded in identifying over 1,000 genes that affected development in some way, about 120 of which were specifically involved in pattern formation. And armed with this information, you can imagine, Nusslein-Volhard and Wieschaus, as well as a number of other researchers, have begun to unravel the details of the specific roles that some of these genes play in development. Now, one of the first mutations that Nusslein-Volhard and Wieschaus described was of a gene they named bicoid, which means two-tailed. They gave this gene this name because the mutant phenotype associated with a mutation in this gene didn't develop any of the structures normally associated with the anterior end of the larva, in other words, the head. What you would get instead were larvae that were essentially two tails attached to each other at the middle, no head at all. Thus, this gene clearly has something to do with head formation. And the question is, what does it have to do with head formation? Well, when Nusslein-Volhard and Wieschaus performed genetic crosses with individuals carrying this mutant gene, they found an interesting thing. They found that bicoid displays a pattern of inheritance we call maternal affect inheritance. What this means is that the phenotype of the offspring, what the offspring turns out to look like, depends not on its own genotype, but instead entirely on the genotype of the mother. In other words, whether or not a mutant larva, or whether or not a larva develops the mutant bicoid phenotype doesn't depend on whether it's inherited the mutant form of the bicoid gene. Instead, what matters is whether its mother has the mutant allele. Now, how can this be? Well, it turns out that the way you explain maternal affect inheritance is this. Bicoid codes for a cytoplasmic determinant. In other words, one of these components that's added to the egg by the mother. Therefore, it's the mother's genotype affecting her ability to add or not add this maternal determinant that affects the outcome of the phenotypic development of her offspring, not the genotype of the offspring itself. Now, if you look at the distribution of the protein produced by the bicoid gene in normal zygotes, you find that in the normal zygote, bicoid is distributed asymmetrically along what will eventually become the anterior to posterior axis of the animal. How does this asymmetric distribution originally arise? It comes about from the fact that in the mother's ovaries, where the egg is developed, there are some cells that produce a large amount of the messenger RNA of the bicoid gene, and then transfer that messenger RNA into the egg. But the cells that produce the bicoid messenger RNA are only located on one side of where the developing egg cell is. So as these messenger RNAs are transmitted or transferred into the egg, they're only transferred into it on just one side. Then when the egg is fertilized and turns into a zygote, these messenger RNAs are translated into the bicoid protein, and what we're left with is an asymmetric distribution of bicoid protein, with bicoid protein being very high concentration on one end, the end that will eventually turn into the head, and very low concentration on the other end, the end that will be the tail. Now you can demonstrate that bicoid is responsible for initially establishing the head-to-tail axis of the fly embryo in several ways. For example, what you can do is actually inject bicoid protein or even bicoid messenger RNA into what should become the tail end of a normal embryo. And in this case, you get an embryo which develops two heads. One that is from its own bicoid protein on the normal anterior end, and one that is the result of the bicoid protein that has been introduced on to the posterior end, where a tail should grow. Now, the question is, what does bicoid actually do that can affect development in this way? Bicoid, as you might guess, is a transcription factor. In other words, bicoid turns on or off genes that are involved in producing head structures. And so, as a result, that part of the developing embryo that has a high concentration of bicoid protein, a high concentration of this transcription factor, will be the end of the embryo where the genes that are involved in producing head-like structures get turned on. But it actually isn't that simple. It's not that bicoid is the one gene that turns on everything that should produce a head. Instead, what bicoid and other similar maternal determinants do is turn on a cascading sequence of other genes, genes that are themselves regulatory genes coding for other transcription factors. You can imagine that many genes have to be expressed in just the right position and at just the right time to develop anything as complex as the structure of an insect head. The way that a single initial factor with an asymmetric distribution, such as bicoid, can go on to coordinate the activities of all these genes is to have a series of additional switches downstream, as it were. Each switch adding more and more specificity and potentially controlling the expression of more and more genes, a cascading sequence of gene expression. Now, to get an idea of how this works, let's look at how the segments of a developing Drosophila embryo are established. The initial gradient of bicoid is responsible for establishing this basic segmentation by turning on a group of genes that we call in general segmentation genes. Actually, bicoid doesn't act alone in this regard. There are some other maternal determinant factors that also play a role, but we can overlook those for the sake of simplicity. There are three classes of so-called segmentation genes. We call these gap genes, pair rule genes, and segment polarity genes. And these three classes essentially represent the sequence of the genes being turned on. Bicoid serves as a transcription factor that regulates the activity of a number of gap genes. The gap genes actually turn out to be responsible for organizing large areas of the head-to-tail axis into basic subdivisions, essentially setting up the initial grid of signals along this dimension. If you have a mutation in a gap gene, what happens is that an entire set of segments fail to develop in the embryo. But the protein products of the gap genes are themselves transcription factors, not actually the proteins that the animal is built out of. These gap genes, then, their proteins, turn on the expression of another set of genes, which we call pair rule genes. The pair rule genes divide the embryo on a finer scale into pairs of segments. If you have a mutation in a pair rule gene, what happens is you have embryos, for example, that will only have half the normal number of segments, with every other segment being lost, hence the name pair rule. Now, the pair rule genes are also themselves transcription factors, which then in turn regulate the activity of another group of these segmentation genes called segment polarity genes. The segment polarity genes are responsible for establishing the anterior to posterior orientation or asymmetry within each section. So if you have a mutation of a segment polarity gene, you'll have all the right number of segments in the animal, but the internal organization of segments can be screwed up in one factor in one way or another. Now, the products of these segment polarity genes are, again, transcription factors which go on to turn on and off other genes. The point here is that what we're seeing in this cascade of gene activation is not the expression of the proteins themselves that the animal is going to be built out of, but instead the expression of transcription factors. And as we go further down this hierarchy, this cascading hierarchy, what we get is an increasingly refined pattern of regulatory gene expression. That is a more complex pattern along the anterior to posterior end of the embryo. And by the time you get to the end of this cascade, what was originally a very simple pattern, that is just a distribution of a maternal determinant like bicoid that's high on one end and low concentration on the other end, has now become a much more complex pattern of different regulatory factors, different transcription factors, having different kinds of concentration gradients throughout the organism that differ from each other. And so if you looked at any combination of these transcription factors at any point along the embryo, you would see different concentrations for different ones of these. Now, to make this point a little bit clearer, let's actually look at one particular pair rule gene. This is called the even skipped gene, or we often call it just the EVE gene for short. Now, if we look at the distribution of the EVE gene in a normal embryo, you actually see a quite complex pattern. You'll see 7 discrete bands of expression of this protein along the head-to-tail axis of the animal. You will see regions where none of this protein occurs, and then you will see a very discrete band where you find this protein. So we have a fairly complex pattern, 7 discrete bands, whereas we started out with a very simple pattern. The question is, how do we get such a well-defined set of stripes from what was initially a less patterned embryo? The answer to this question comes from looking at the distribution of gene products that were expressed earlier in this regulatory hierarchy and considering how these gene products collaborate as transcription factors. Now, to think about this, I want to remind you of a discussion we had earlier when we were talking about gene expression in eukaryotes. Remember that gene expression in eukaryotes involves regulatory regions that precede the structural gene, regions where the transcription factors that will turn on and off that gene must come down and bind before RNA polymerase can go on and do its work of transcribing that particular gene. Remember, any particular gene will often have many transcription factors that have to bind in the right combination in order for that gene to be turned on. Now, the interesting thing about the EVE gene is that if you look at its regulatory region, it's huge, and in fact it has 7 independent sets of regulatory sequences, each of which responds to a different combination of regulatory proteins, a different combination of transcription factors. And this is an important point. If any one of those regulatory regions has its appropriate concentration or set of transcription factors, then the EVE gene will be expressed. So we essentially have 7 kinds of locks, or seven sets of locks. If any of those locks is unlocked, then the expression of that gene will occur. You don't need to have more than one of these regulatory regions turned on, just one will do. Now, how does that help us understand where we get segmentation from, or the appearance of these very discrete stripes of the EVE gene? Well, imagine that you're measuring the concentrations of different transcription factors that have arisen from this regulatory cascade of regulatory proteins along the head-to-tail axis. So you have a little device that allows you to go, say, from the head end to the tail end, and at any particular point along that axis say, what is the concentration of a particular set of transcription factors at this point along the axis? What you'll see is that at any one point, the many transcription factors that are now all in that developing embryo will be at different concentrations relative to each other differently along that axis. In other words, one might be going down as you go from the head to the tail. The other one might be increasing in concentration as you go from the head to the tail. Another one might be in high concentration in the middle, but low concentration on both ends, with increasingly complex patterns as you move on down this hierarchy. The way you get this complex pattern emerging is that if a particular regulatory sequence needs just a combination of these genes, then at the point along the embryo where the right combination of concentrations of transcription of factors occur for that regulatory region, where that occurs, the gene will be turned on. But as you move farther down, the relative concentrations of transcription factors may now change so that regulatory region is no longer finding its right transcription factors and the gene is turned off. As you move further down the embryo, a different set of concentrations might turn on a different regulatory region for the same gene. And this is the way that you get the emergence of pattern from an initially unpatterned embryo, as you have many, many regulatory factors, many transcription factors being expressed in this cascading hierarchy. Now, let's make this mechanism clearer a little bit by just looking briefly at one regulatory sequence and see how it reads the positional information. If, for example, we were going to look at just the second band of EVE expression along the axis of the animal, or what we might call EVE stripe 2, we would expect that there would need to be particular concentrations of, to simplify the model, 4 regulatory proteins. There's a protein, the original protein bicoid, that it needs to see, it needs to have to turn on expression. It needs another regulatory protein called hunchback. And it also needs to not have 2 other regulatory proteins that would repress expression. These are called giant and cripple. These are somewhat fanciful names that come from the nature of the phenotype of mutations that arise in these genes. At only a particular point along the axis of the animal will you find the particularly correct high concentration of hunchback and bicoid and low concentration of giant and cripple. And it's only at that one point along the axis where EVE will be expressed by having its regulatory region corresponding to the second stripe turned on. Well, with the expression of segmentation genes, what we've done is been able to define the basic segmental structure of a Drosophila embryo with a polarized anterior to posterior axis. And as I said, in later phases of development, these segments will go on to produce very different kinds of structures depending on where they are. The differentiation of these different segments to produce these different kinds of parts is actually mediated by the activity of yet an entirely different set of genes called homeotic genes. Once the segmentation has been established in a fruit fly embryo, homeotic genes then are essentially the master regulatory switches that will turn on the development of specific structures appropriate for that segment. Now, the interesting thing about homeotic genes the interesting point to make about homeotic genes, and in fact about developmental genes or developmental regulatory genes in general, is that if you look at the structure of these developmental genes across very different kinds of organisms, you find that these different organisms sometimes share remarkably different or remarkably similar sets of genes. For example, there's a set of genes in vertebrate animals, things such as chickens, mice, or us, that are also involved in determining the specificity of how parts develop along the anterior to posterior axis. We call these Hox genes in vertebrates that are remarkably similar in their structure, that is, in the sequence of DNA, as to the homeotic genes that specify different parts along the anterior to posterior end of a Drosophila. Remarkably similar, so much so that if you have a Drosophila embryo that has a mutation in a particular homeotic gene, for example, a homeotic gene that would be associated with producing something like an antenna, you can find what we would call homologous gene in a chicken that you could put into that Drosophila embryo, and then with that chicken gene, the Drosophila embryo would go on to develop correctly. This is remarkable because insects and vertebrates haven't had a common evolutionary ancestor for hundreds of millions of years. Yet we have this enormous similarity in the genes that are controlling basic developmental mechanisms. This similarity, such as the similarity between homeotic genes in insects and Hox genes in vertebrates, illustrates a deep commonality in the basic developmental genetic pathways that animals use when they develop. And it essentially suggests that there's some sort of fundamental genetic toolbox that evolved very early on. Not the specific genes that control the specific details of how animals look, but instead those master controller genes that start unfolding the basic pattern and that are themselves responsible for turning on the more specific genes. And it's this genetic toolbox that has been used to more or less create all the varied body plans of animals that subsequently arose in evolution. Well, we've only really scratched the surface of looking at how the patterns and processes of development work, especially things like the emergence of complex patterns such as the heads of insects. Our example of Drosophila serves as a good model, at least as a first approximation, for understanding how genes, how the environment on several levels can interact and inform each other as a multicellular organism develops from a single cell. But now that we've looked in some detail at how these multicellular organisms develop, we're left with another set of questions. Specifically, how do all the parts of this organism coordinate their activities so that they can function smoothly as an integrated unit? And this is the question that we're going to begin to turn to next time.